Hello grade 8 students. In this video we're going to talk about digestion part 4, and in specific we're going to talk about enzymes. So first let's remember some notes about the digestive system. Digestive system is divided into digestive tube and digestive glands. Digestive tubes, organs through which the food passes, whereas digestive glands, organ through which food does not pass. You have to know that the organs of the digestive tube includes the mouth, esophagus, the stomach, small intestine, large intestine, and finally we have the anus, where the excess food and the ways they are going to be excreted. Whereas we have different digestive glands. We have salivary glands that are found in the mouth, gastric gland in the stomach, liver and pancreas, they are about the small intestine level, and we have finally the intestinal glands. So let's start. What is the role of these glands? Digestive glands, they secrete digestive juices into their site of action, meaning that they secrete juices into their place. Where do they work? Where do they act? They secrete juices. Let's move to know more. Here we have the table showing the digestive glands, the juice that is secreted by this gland, and their site of action. So salivary glands, what do they secrete? In the mouth, they secrete the saliva. Where the site of action is the mouth, where the medium, it's neutral. So the pH, it's around 7. It's approximately 7. It's not exactly 7. Maybe it's 6.5 to 7. Okay? So here the medium is neutral inside the mouth. While the gastric gland, they secrete a special juice. It's called the gastric juice, where into the stomach, and the medium is acidic, meaning that the pH, it's less than 7. 6, 5, 4, 3, 2. So as we move from 7, the acidity will increase. Whereas pancreas secretes pancreatic juice. Liver secretes bile. Intestinal glands, they secrete intestinal juice. All of them, their site of action is the small intestine, and the medium is basic. So medium is basic, meaning that the pH, it's greater than 7, 8, 9, 10, 11, 12, 13, and 14, it's the end. So here we're talking about the pH scale. So let's move to talk about neutral. It's around 7. 7, it's 7, 6.5 to 7, even though 7.5, it's considered as a neutral. Whenever it's pH greater than 7, so here we're talking about the basic. So as we move away from the 7, the basicity, will increase. So it will be more basic. So the strongest base is the one that is away from 7. It's not 9 or 10 or 11 or 12. It's 14 is the strongest base. While the one that's near to the 7, it's said a base, but it's a weak one. While regarding the acidic, so here pH, we said it's greater than 7. While here in acidic, the pH, it's less than 7. It's 6, 5, 4, 3, 2, and 1. So as we move away from the 7, the acidity increase. While here, as we move away from the 7, the basicity increase. So the strongest acid, it's here, while the strongest base, it's here. And these are examples about the pH. For example, the gastric juice in the stomach, pH equal 2. Tomato juice, the pH is equal to 4. It's also acidic. Human urine, the pH is equal to 5. So it's also acidic. While pure water, the water that we drink, it's in neutral. pH is equal to 7. 7.4, it's the human blood. It starts to move away from the neutral, to move to the base. Soap that we use, hand soap or gel or the shampoo and so on, So the pH equal to 10, so it's basic, while the household bleach, its pH is equal to 12. So this is the strongest base here, and here are the strongest acid. So as we move away from the 7, either we are moving to move to the strong acid or strong base. What is digestion? Digestion is the set of both chemical and mechanical transformation. Mechanical is cutting the food into smaller pieces, while chemical is the action of the enzymes in the juices. And we are going to talk about the enzymes. So it's the set of both chemical and mechanical transformations of complex food molecules into simple nutrients to be used by the body cells to produce the energy needed for our activities. So complex food is going to be digested into nutrients to be used by the body cells to produce the energy. So this is the definition of digestion. Keep in mind before we start, all the digestive juices, all of them, what do I mean by these digestive juices? Digestive juices, here I mean by, for example, we have the saliva in the mouth. So this is one of the digestive juices. So we have also the gastric juice in the stomach, pancreatic juice, the bile, intestinal juice. All of them, they contain enzymes except for bile. Bile is the only juice, which is secreted by liver for sure, is the only juice that contains no enzyme. So what are the enzymes? Enzymes, they are like a scissor. They cut, they break down, they digest, they transform, they hydrolyze the food into smaller elements. So enzymes, they are called the biological scissors. So the scissors, and it's in the biology, so it's called biological scissors. It digests, it breaks down, it cuts, it transforms, it hydrolyzes. All of them, they have the same meaning, digestion of food. So they are called the biological catalyst. What is the function of the catalyst? Catalysts, they speed up the reaction, they accelerate the digestion process, they make it faster, but they remain unchanged. So they stay the same. For example, if I have 1 milliliter of a specific enzyme, this enzyme will stay the same whenever it finishes the digestion process, but it will accelerate the process, it will make it faster. So enzymes, they are important to accelerate meaning that to make it faster, to speed up the reaction of digestion. Let's move to this table. This table is a very important table. We're going to divide it into two parts. The first part in this session, and the next session we'll move to the second part. So in this table we have the organ, we have the digestive gland, the name of the gland, the name of the juice, the name of the enzyme, substrate. What the meaning of substrate? For example, this enzyme works on which type of food? So this type of food is called the substrate. For example, it works on protein, so protein is the substrate. It works on lipids, so lipids is the substrate. It works on starch, so starch is the substrate. The product, meaning that the end result of the digestion. So the result of the digestion, what is the final result of digestion is called the product. Chemical medium, whether it's acidic, basic, or neutral. Temperature, it's 37 degrees Celsius, always and always. So we have the mouth. In the mouth, we have a special gland. It's called the salivary gland. What does it do? It secretes saliva. The saliva contains a special enzyme. It's called salivary amylase. This salivary amylase will work on starch, and starch is the substrate and is digested to give maltose, which is the end product, and the medium is neutral, meaning that the pH is approximately 7 and the temperature is always 37. This is the normal body temperature, and the digestion will occur at this temperature, 37. So I repeat, the mouth contains a gland, salivary gland. It secretes a juice. It's called saliva. This juice contains an enzyme. It's called salivary amylase. Salivary, since it's found inside the saliva. The saliva is the one that's found in our mouth. This is the saliva. And it works on which substrate? On this starch, to give which product? The maltose. The medium is neutral. In the mouth, always the medium is neutral, and pH equal approximately to 7, and temperature 37 degrees. This is the saliva. The saliva that's found inside our mouth that help us to digest the food. So this is the saliva. Let's move on to the second organ, the stomach. In the stomach, we have a special gland. This gland is called the gastric gland. This gland secretes a special juice. It's called gastric juice. This juice contains an enzyme. It's called pepsin. Pepsin, it works on which substrate? It works on protein. To give which product? It gives the peptide, the polypeptide. Peptide. The medium is acidic, meaning that the pH is less than 7. And the temperature always 37 degrees Celsius. So how can I study pepsin works on protein to give peptide? Pepsin, it starts with the letter P. Protein starts with the letter P. Peptide, so it starts with the letter P. So we have P. So pepsin, protein, peptide. So always, whenever you have to remember this, pepsin, P, works on protein, P, to give peptide, the 3 P's. Pepsin works on protein, P, to give P, which is the peptide. So the 3 P's, here we know that pepsin, it works on protein to give peptide. Mister, how can I know that pepsin, it works in an acidic medium? Wait a minute. Pepsin, it's similar to which word? It's similar to the word Pepsi. Pepsi, it's an acid. You drink Pepsi, all of you. So Pepsi, it's an acid. So pepsin, it works in an acidic medium. These are the only 2 differences in the medium, mouth and the stomach. And the rest, all of them, they work in basic, and we will see. So here, the pH level of the stomach. The stomach is the pH is about 4 or 5. So it's less than 7. So it's acidic. Moving to the small intestine. In the small intestine, we have the pancreas. Pancreas secretes a special juice, which is called pancreatic juice. Pancreatic juice, it contains an enzyme called peptidase. Every word that ends with ase, from now on, it's an enzyme. So peptidase, enzyme that digests peptides. Peptides is the intermediate form of protein. So it will give what? Amino acid. The medium is basic, meaning that the pH is greater than 7 and temperature 37. Here it's very easy. From its name, peptidase, it digests peptides to give amino acid. So this is the simplest form of protein. Okay, so and the peptide, they are the intermediate. So pancreas and pancreatic juice, and this is the pancreas, and this is the called pancreatic duct. Moving to the second juice, also we are talking about the pancreas. So pancreas secretes a juice, it's called pancreatic juice. Here we have an enzyme trypsin. Trypsin, it works on protein to give amino acid. So this is protein, this is the complex form, while amino acid is the simple form. So here it digests immediately the complex into the simple, and the medium is basic, meaning that the pH it's greater than 7 and the temperature for sure is 37 degrees Celsius. So here you can conclude that Trypsin is very strong. It digests protein immediately into amino acid. Whereas if you notice here, pepsin, it digests protein into peptide, the complex form to the intermediate form, and not to the simplest form. So which one is stronger? Sure, trypsin is stronger here. So trypsin also works on a protein, but it gives amino acid. So it's faster digestion from the complex to the simple. While here in the case of pepsin, it works from protein to peptide. Moving to the third enzyme inside the pancreatic juice. Pancreas also here, you see pancreas, and also we move to the pancreas. So pancreas secretes pancreatic juice, contains an enzyme called amylase. Wait a minute, amylase is similar to this? Yes. They have the same function? Yes. But here it's called salivary amylase. So it's found inside the mouth. So the medium here is neutral, while the amylase is found in the pancreas. Also it works on starch, the same, and it gives maltose, the same, but here the medium is basic, meaning that the pH is greater than 7. So here both of them, they are the same. They have the same function, but the difference that the salivary amylase, the pH is in neutral. So medium is in neutral, so pH is equal to 7. While amylase in the pancreas, it works on starch also, to give maltose also, but the medium is basic, meaning that the pH it's greater than 7. So here, similar to each other, amylase and salivary amylase, they have the same function, but they differ in the medium. Salivary amylase in the mouth, so medium is neutral. Moving now to the last in the pancreatic juice. Pancreas secretes pancreatic juice that contains the enzyme lipase. As I said, ase from now on, ase it's meaning an enzyme. So lipase, from its name, lipase, it digests lipids. So the complex form of lipids is the lipids, the fats. There is no intermediate. We have the simplest form, which is called fatty acids plus glycerol. The medium is basic, meaning that the pH is greater than 7 and the temperature is 37. As you see here, all of them, they have a basic, unless the gastric juice and the stomach acidic and in the mouth neutral. Okay, so I repeat, pancreas secretes pancreatic juice that contains the enzyme lipase. Lipase, as its name indicates, it works on lipids to give what? There is no intermediate. Always we have simple form, which is the fatty acids plus glycerol, and the medium is basic, pH greater than 7. The last gland, it's the liver. Liver, it works on. So liver contains a juice which is called the bile. Remember what do you say? That liver contains a juice is called the bile. Bile is the only juice that contains no enzyme. So here we have no enzyme. So it works on lipids to give emulsified lipids. What the meaning of emulsified? We will see. pH, it's greater than 7 since the medium is basic and the temperature 37. What the meaning of emulsified lipids? Here we have the lipids or the fats. Emulsification meaning that we have to separate them. So bile, it works on the separation. We have large fat, so it becomes smaller fats, and then the lipase will work to digest it. So the bile is going to separate the large molecules. So it's going to degrade the large one into smaller ones. That's why digestion is going to be easier by the enzyme lipase that digests the lipids into fatty acids and glycerol. So the role of the bile is the emulsification. So separation of large and complex food molecules into smaller one. This is the role of bile. So bile has an important role in the emulsification. Emulsification meaning that's the separation or degradation into smaller pieces, or cutting them into smaller pieces. Then lipases or lipase will work on lipids and digest them into fatty acids and the glycerol, which is the simplest form of the lipids. Thank you, guys. Goodbye.